Hey guys, so today is Wednesday, January 27th, and today we are covering organelles. Okay, so a couple of things that I want to remind you about. Okay, so Wednesday, January 27th, the day that you should be doing doing these notes and completing these notebook pages. But if you are working ahead of time, just know that Thursday, January 28th, you have your Unit 3 test for me. Okay, so your Unit 3 test is going to be on Thursday, January 28th. On Thursday, you have to take your Unit 3 test. You have to turn in your Unit 3 notebook. You have to turn in your microscope lab is officially due that day if you haven't already finished it from Monday, and then your organelle discussion post. Okay. Now, I went ahead and uploaded the Quizlet for y'all, so y'all can go ahead and start studying with that. I'll show you how to access the Quizlet in just a second. But I just wanted to go ahead and give y'all that reminder. Okay, so I'm gonna start sharing my screen with y'all so you can, so I can show you where the Quizlet is. Okay, so if you go to your Schoology course page and you go to Unit 3, okay, and scroll down, you'll see study materials for Unit 3. If you click the Unit 3 Quizlet, not the chain link, you have to click the words itself. It's gonna open you up, but see how it says Quizlet refused to connect? That's fine. Forget about this. You just have to click the word Unit 3 Quizlet one more time, and then it takes you to the Quizlet page. Okay, so then you can do flashcards, learn, write, spell, test, okay, whatever. I do the flashcards and I do the matching, okay, or the testing is really easy. And that's just there's only 33 note cards. And basically, what I've done for the Quizlets is taken your notes and the notebook pages and combined them and made like a little study sheet for you. Okay. So I'm going to go back to today's folder. Okay. So this is today's folder, January 27th, organelles. Okay. This strange teaching video, that's the video you're watching right now. Okay. So now I'm going to cover what you should be doing today. All right, so first up, of course, the Amoeba Sisters, okay? So this Amoeba Sister video is going to walk you through the definition of all of the organelles that I'm gonna cover today, okay? So go ahead and watch that. There's 3 videos here. I really just want you to for sure watch the first one. And then when you're studying later for the test and you don't wanna look at Quizlet and you don't wanna look at your notes, go ahead and watch these other videos as well. Okay, so I'm going to give you a minute to pause my video and watch the Amoeba Sisters. Okay, so now I'm assuming you've watched the Amoeba Sisters. So if you scroll all the way down, okay, this Organelles 8.2 PPT, that's the PowerPoint notes that we're going to cover today. All right, so if you want to download them and split screen, great. If you would just want to leave my video pulled up and pause it at each slide, you can do that too. Okay, so let me get this opened up. All right, so cell structures. Okay, so cell structures equal organelles. Okay, that's what I want you to title this. So cell structure is organelles. So that's what you're going to title your notes for the day is this top part. Okay, so now, moving on. So animal versus plant cells. So this is the last slide from our notes from yesterday. Okay, so a couple of things that I want to go over. So what's the big difference between an animal cell and a plant cell? Well, first of all, their shapes are different, right? So animal cells are going to have lysosomes and centrioles, okay, where the lysosomes are going to be these purple, like kind of like disc objects that are right here, and then the centrioles are going to be these like spindle fibers, okay? And then plant cells are going to have the cell wall, chloroplast, and then this large jelly bean vacuole. So you can look at them and tell, okay, there's this big jelly bean here. This is obviously significantly different than not having a jelly bean in this or in this plant cell, animal cell, excuse me. Okay, so you don't have to write anything for that slide if you already have this written down. If you don't, go ahead and rewrite it. Okay, so first up is the cell membrane or plasma membrane. Okay, so I'm going to change this. I want you to write cell membrane equals plasma membrane. This is um, this is an interchangeable word, so they will use both of these words on the test. So they'll say the cell membrane consists of what? And then they'll turn around and ask you the plasma membrane consists of what? It's the same thing, okay? It's just same word, same meaning, different word, okay? So the plasma membrane is going to control what comes in and out of the cell. Okay, so that's your first bullet point for this, and it's made up of lipids, proteins, and carbohydrates. Okay, if you just want to write carbs, there you go, abbreviate it. But basically what this is your lipids are going to be your little octopus guys. So that's that little red top hat, and then you have the little tails are going to be the octopus feet. So I call my lipids like my little octopus guys in the plasma membrane. Your proteins are going to be these channel proteins or integral proteins or peripheral proteins. We'll get into those in a later unit. Okay, just know that there are proteins in the plasma membrane. And then the carbohydrates are going to kind of form like little chains off of the proteins. Just know, oh, that says, okay, so just know that they are going to be on top of the proteins. Okay, so cell membrane. This is getting, what I want you to write on this slide is we're going to get into the structure of a lipid in a cell membrane, okay? Because I'm going to tell you all about the proteins later, but I really want to talk about this lipid, okay? So I want you to, I'm going to zoom in. I want you to redraw this little lipid octopus guy, okay? So redraw the octopus. So the top part is hydrophilic, okay? So hydro is talking about water, right? And philic is loving. So hydrophilic means that it likes water. So the little orange, not orange, the yellow part of the octopus, okay, is water loving. So I want you to redraw this image. So the top part, hydrophilic head equals water loving, okay? So then the little tail part is hydrophobic, phobic meaning hating. So it hates water. So the tail hates water. So you have to remember the order of this, that there's a hydrophilic head and a hydrophobic tail. So the way that I remember it is philic over phobic. So Mr. Phil puts his top hat on. So the hydrophilic, so the water loving head of the lipid is opposed to the hydrophobic, the water hating tail. So philic, okay, Mr. Phil's top hat, head, water loving. Okay, so after you redraw that, that's what I want you to take away from this slide. Okay, so the function of the cell membrane and all of these proteins and lipids is to regulate what enters and leaves the cell. So I do want you to include this, regulate what enters and leaves the cell. Okay, this is how materials can enter the cell and then how waste can leave. Okay, so after you get that, on to the next one. So now these are basically going to be definition after definition of organelles and parts of the cell. Okay, so if you, I want you to take down these notes, but it would also be super speedy for you if you go ahead and get out your interactive notebook and flip to page 4. So then it's going to be on pages 4 and 5. So then whenever you get to this organelle, you could write it in your notes and write it in your notebook, and then you're done. Okay, so cell wall is the rigid outer layer part, outer layer of plant cells. It's also found, so I just want you to write cell wall equals rigid outer layer. And then I also want you to say that it's in plant cells, bacteria, fungi, and some protists, okay? It is made up of carbohydrates, but each cell wall is going to be made up of different types of carbohydrates. Okay, so like some plant cells have chitin in it, and some are made up of cellulose. So it's gonna depend. All right, so I just want you to have cell wall, rigid outer layer. It's found, examples of this are plant cell, bacteria, fungi, and protists. Okay, so a couple of things that I want to write here is that see how both cells are going to have a cell membrane. Okay, so this is, what type of cell is this? This is going to be an animal cell, okay, because here, it's a bad picture, but this is my jelly bean. So this is an animal cell, plant cell, and a prokaryotic cell, okay? So all cells have a cell membrane. See this arrow, how it's pointing to the animal, the plant, and the prokaryotic cell, but only 2 of them have a cell wall, okay? So cellular boundaries, I want you to write all cells have cell membrane. What's another word for cell membrane? Plasma membrane. Okay. And then I also want you to write all cells. Let me move my picture. All cells do not have cell walls. Okay. So after you get that, you're good for this slide. Okay, so cytoplasm is the jelly-like fluid that fills the cells. So this is the gel-like fluid where organelles are found. I want you to write jelly-like fluid that fills the cell. Okay, so basically this is going to be like the air inside of the cell is going to be filled with this gel-like fluid called cytoplasm. Okay, so then we have our nucleus. Our nucleus is going to control most of the cell's processes, and this is where your DNA is going to be found. So nucleus, first bullet point, controls most of the cell processes. When you say contains DNA, so chromatin equals condensed DNA. Okay, we're going to get to that on another slide, but if you want to go ahead and write that down, you can. Okay, so this is your, this purple structure is representing a nucleus. Okay, you can see it in the animal cell and the plant cell. So what type of cells have a nucleus? Prokaryotes or eukaryotes? Eukaryotes, okay? So eukaryotes have a nucleus, and this is an example of it. So this big purple thing as a whole is the nucleus. So now we're going to look at the organelles technically inside of it. Okay, each little hole that kind of looks like a blackhead pore, those are nuclear pores. And that's just how proteins and other information can like get into the nucleus and how it can like send information out. Okay, chromatin is going to be that condensed DNA. So it's like all of your super packed and organized DNA. Your nucleolus is going to be like the dead center of the nucleus. This is like where the control center is. And then this nuclear envelope is like the protecting layer of the nucleus. You don't have to know all four of these. I just want you to have this written down up here. Okay, so nucleolus is found inside the nucleus and produces ribosomes. So nucleolus found in nucleus and produces ribosomes. Okay, so these ribosomes are important because the ribosomes are going to form other proteins, which can make more DNA, or they can go and have a specific function. Okay, so then we have our chromatin. It's tiny strands inside the nucleus that contain instructions for directing the cell's function. So I just want you to write instructions for directing cells function, and then it's condensed DNA. Okay. Okay, so the endoplasmic reticulum, so it's going to look on a well, you'll see it whenever I explain it in the notebook when you can like see the different types of organelles. But to me, the endoplasmic reticulum looks like on a TV commercial when you have like the most perfect deli like sandwich and like how the deli meat is like perfectly folded. So that's kind of what the endoplasmic reticulum looks like. And it's always going to be connected to the nucleus. It's going to be like super close to the nucleus because the nucleus is feeding it the ribosomes that it sends out to the rest of the cell. Okay, so to clean this up and to make this pretty in our notes, I want you to write that the endoplasmic reticulum equals passageways that carry proteins and materials from one part of the cell to the other, okay? And then we have two types of endoplasmic reticulum. So write the whole word endoplasmic reticulum out and then put in parentheses ER, just like how it is up here in the title. So then that way you don't have to write the whole word out over and over again. Okay, so this deli meat, it's gonna have two types of it. Okay, so I'm gonna blow up this picture just a little bit for right now. Okay, you can see this is the rough ER and this is the smooth ER. The only difference is the rough ER has these polka dots in it. I call it the pepper on my deli sandwich. These polka dots are called ribosomes, okay? And that is basically what the nucleus is producing, and the ribosomes are going to help assemble more amino acids down the line and create more proteins. Okay, so that's kind of what it looks like. So the difference, so both of the rough ER and the smooth ER are going to be carrying proteins and other materials in and out of the cell so it can then specialize. But the rough ER produces proteins and has the ribosomes on it. So rough has ribosomes, smooth, no ribosomes. So rough equals produces proteins equals ribosomes on it, where the smooth ER produces hormones and lipids, okay, because it's not producing ribosomes. So no ribosomes on it. So I want you to like underline that, bold, okay, no ribosomes on smooth. Okay, so if you want to write like the salt, pepper, I mean, not salt, pepper, the turkey meat and like pepper analogy, feel free, go for that. Okay, but I wanted to make sure that you for sure have this down. Okay, the other thing from this slide that I want you to have written down is that ribosomes, okay, they're going to assemble amino acids to create proteins. Okay, you can find them on, so you can do a weird arrow underneath rough ER and in the cytoplasm. So the ribosomes are going to be just kind of in the cytoplasm. What's the cytoplasm again? It's the jelly-like fluid that fills the cell. So it's going to be in the rough ER and then just kind of like free floating around the cell. Okay, so just like on the last one, how I told you that the endoplasmic reticulum kind of looks like deli meat, the Golgi apparatus is going to be the same way. The way that you can tell them apart is that the Golgi apparatus is not going to be super close to the nucleus. The ER, the rough ER and the smooth ER are going to be super close, like almost touching the nucleus, where the Golgi apparatus is not, okay? So the Golgi apparatus is kind of like the UPS like the Amazon warehouse, okay? So the Golgi apparatus is going to receive proteins and materials from the ER, package them, and distribute them using vesicles, okay? So let me flip my page. Okay, so we're going to write this whole thing down. So the Golgi apparatus, instead of writing body complex, I want you to write Amazon warehouse, okay? Because think of it as, so the Amazon people are receiving this protein. So they're receiving your hand sanitizer that you just ordered, okay? And they're receiving it from the ER, which is the endoplasmic reticulum, so from the company. And then the Amazon warehouse is going to package them into their prime shipping container and then distribute them to the prime trucks or the UPS trucks. So these UPS trucks or prime trucks are these vesicles, okay? So write down this definition, and then I also want you to write down that the vesicles equal UPS truck. Okay, so we're gonna, I'll use this analogy over and over again. And the next 3 slides are basically like an overview of this process, okay, where I'm gonna go into it in a deeper detail. Okay, so this is the like part 1 of the process. See, I'm gonna fast forward. Okay, this is part 2, and then part 3 is the whole picture. Okay, so now I'm gonna explain each part. Okay, so first up, let me zoom in. So this is your nucleus, okay? So then if it's the deli meat that is touching the nucleus, it has to be your ER, right? So this is my ER. So then if it has ribosomes on it, so these little polka dots, it has to be what type of ER, rough or smooth? Rough. Okay, so my proteins are assembled on ribosomes. So they come from the nucleus. The ribosomes and proteins are assembled together, okay, and then they complete their way through the rough ER. So the ribosomes and proteins have now made their way through the rough ER, okay. Now proteins are carried to the Golgi apparatus using vesicles. So see how like this is the rough ER, it's now gone into the smooth ER. So this is the cytoplasm of the cell, so the jelly-like fluid that fills the cell. So now in order for it to leave the ER, it can't just like shoot into space. Otherwise, it's not going to know where to go. Like if you just throw an Amazon package out into the middle of the road, it's just going to get run over. You have to have that UPS driver deliver it to your house for it to actually make it there. That's why you have to use this vesicle. Okay, so the proteins are then put from the ER into a little vesicle, and now it can go to the Golgi apparatus. Okay, so here's the little vesicle. It's now driving to the Golgi apparatus. It makes it there. There's my deli meat that's not connected to the nucleus. So see, the nucleus is usually a purple color, like in this diagram, and see how it's not there? So that's how you can know that this is the Golgi apparatus and not the nucleus. Okay, so this is where the Golgi is going to sort the proteins and package them and basically rearrange them and distribute them, where then the vesicles, so now the UPS truck has now sent it to their final destination. So the Golgi apparatus is kind of like how all of our Amazon packages go to Memphis, and then Memphis is like, okay, these ones are going to South Haven, these ones are going to Hernando, these ones are going to Collierville. So then the little South Haven UPS truck will deliver them to us. So that's kind of what the Golgi apparatus does. It's kind of like the big warehouse that is like the distribution center. Okay. So this is like the overview. I would honestly like print screen this image. It's slide 17 out of 28 on this PowerPoint. So if you wanted to print this out or redraw this slide is a great tool. I actually printed it out and put it in my notebook. Okay, so almost done. Okay, so vacuoles store food, water, waste, and other materials. So it's larger in plant cells because it has this central vacuole. So I want you to write vacuole equals stores water, waste, and other materials. Okay, so the contractile vacuole, I don't need you to know that it's in paramecium, but that's used to get rid of water. Okay, and then the central vacuole is in plant cells. So let me erase this. Okay, so this is what I also want you to write. So that central vacuole, it looks like a jelly bean. You'll know what I mean by that in just a minute, but go ahead and write that down. Okay, so the contractile vacuole, so this is like in animal cells, this allows it to expel food, waste, and other materials, where the jelly bean, the plants soak up more water than we do. So they're gonna need this big central vacuole in order to get rid of water and store extra water. Okay, so that's so you can't really tell in the microscopic images, but like in the like basic learning organelle images, you can tell it kind of looks like a jelly bean. Okay, lysosomes. Lysosomes are in animal cells only. Okay, so that's important. I want you to write that down. And it's an enzyme basically that breaks down food and other worn out parts of the cell. So basically, if you have an old part of the cell that is like damaged or it's too old to function and it's not working at like the best capacity anymore, the lysosome is going to eat it away so it can regenerate itself and it can start fresh. Okay, so cytoskeleton consists of proteins, filaments. So protein filaments, two examples of that are the microtubules and microfilaments. Basically, these are used for cell shape. So cytoskeleton equals for cell shape. And then I want you to say consists of protein filaments, and examples of those protein filaments are microtubules and microfilaments. Okay, so you don't have to write anything for this slide, but this is an example of what microtubules look like under a microscope. So basically, they're what is going to play a really important role in maintaining the cell shape during cell division. Okay, so this is like when during mitosis and meiosis, when you're like pulling the DNA chromatids apart, your microtubules are going to keep everything in place. Okay, cytoskeleton. So for cytoskeleton, this this slide, yes, it is a part of the cytoskeleton, but I also want you to say that right now we're going to talk about cell movement, okay, like how cells can move on their own. Okay, so I would rather you say cell movement. And the first type that we're going to talk about is cilia. So cilia equals tiny hair-like projections outside the cell, and I want you to know that it is used for slow movement. Okay, so let me bold that for you. Okay, so that's what I want you to write down. So cilia is going to be like these little whiskers that are on the outside of the cell. It kind of allows it to just kind of float with the ocean, like going with the waves, kind of like a flat fish just going with the current. And it's just for very slow, like gradual movement of the cell. Okay, but then if a cell wants to be really fast, okay, so this is where I want you to put number 2 for cell movement. Okay, is flagella. So flagella is going to be a long whip-like structure outside the cell. I want you to say that it's like a tail. Okay, so it's like a tail, and this is used for fast movement. Okay, go ahead and put your example for flagella and flat and fast movement is sperm. Okay, it uses the tail to swim super fast, kind of like a propeller. So I like to think like a flag waving in the wind, like a tail. Okay, so it uses a really fast propeller to swim, where in cilia it's very slow movement. So flagella, fast movement; cilia, slow. Okay, cytoskeleton, this is going to be, so this is continuing cytoskeleton structures. So this is a centriole. Centrioles are used in movement of chromosome during mitosis and meiosis. Okay, so I want you to say centrioles equal used in movements of chromosomes during mitosis and meiosis. Okay, and that it's in animal cells only. Okay. Do, 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 do. All right. So chloroplasts capture energy from sunlight and convert it into food energy, food that contains chemical energy. Basically, what this does is that the chloroplast is like I like to call them the money of the plant. They go up and they capture sunlight, and then they're going to convert it into food, which what food do plants consume? Glucose. Okay. So they're going to eat that glucose, and then it's going to break that glucose down into a chemical energy that the plant can actually use for cellular processes. And so chloroplasts are going to actually contain their own DNA because they're able to go and do all of this. All right. So it's kind of, they're like a little organ. They're like a little cell within a cell. They're kind of just working super hard in order to make energy for plant cells. Okay. So chloroplasts equal capture energy from sunlight and convert it into food that contains chemical energy. That is what I want you to write. And I do want you to also include that they contain their own DNA and that they're found in plant cells. Okay, because I think that this is a test question that she tries to trip you up on, where she will say, which of the following is only found in plant cells? And she'll list like lysosome and nucleus and all of these other organelles. And one of them is chloroplast, and chloroplast can only be found in plant cells. Okay, last slide of notes. So the mitochondria, everybody's always heard of mitochondria, powerhouse of the cell, but what does that even mean? So the mitochondria converts chemical energy from food into compounds that the cell can actually use. So basically the powerhouse, so is converting chemical energy into food. So the mitochondria is taking this energy that the cell is like making, so the ATP and ADP and all of that good stuff, and is changing it into food so that your cells use so that they can complete whatever job that they are trying to complete. So they're also going to contain their own DNA, but they're going to be found in plant and animal cells. Okay, so first thing got to write is mitochondria equals converts chemical energy from food into compounds that the cell can use. Another word for this is cellular respiration. Okay, they contain their own DNA and they are found in plant and animal cell. Whoops. Okay, so that is it for the notes for today. All right. So, couple of other things that I want to do with y'all. All right. So let me exit out of the notes. Okay, and go back to Schoology. All right. So if you go back to Schoology, there's this interactive cell structure activity that you can do. So I'm gonna click it and open it up and kind of go over it with you just briefly here. Okay, so this is the cell structure. So these little arrows down here, so you're just going to click through them and then you're going to answer questions about prokaryotic and eukaryotic cells. You can click the little drop down arrows, answer the questions. Okay, they're not for a grade. This is just an extra great study resource. Okay, so going back. Okay, so today what you had to do was take notes, answer this organelle discussion post. So go ahead and do your organelle discussion post if you haven't already done so. Okay, and then I'm going to go over the interactive notebook pages for today. All right, so these, so today's pages that you had to do were 4, 5, 6, 7, 8, and 10. Okay, I know that sounds like a lot, but it's not, I promise. So all page 4 and 5, okay, I'm going to show you this 4 and 5, okay, are just you're telling me what the definition of this organelle is. So what is the definition of a cytoskeleton? It helps with the shape of a cell. It consists of protein filaments, which are those microtubules and microfilaments. Okay, that's what I want you to write here. Pictures of an organelle. If you're doing this on the computer, go ahead and just Google picture of microtubule, picture of microfilament, and insert that screenshot or image right there. If you're doing it on paper, you know that I am the worst drawer in the whole world. It's fine. Just draw me what you think looks like the cytoskeleton, okay, based on what you learned from your notes today, okay? Obviously, if your picture is way off, I'm not going to count it for corrects for completion, okay? But just use your best judgment, okay? You'll be fine. So that's page 4. Page 5, you're doing the same thing, okay? So page 6, I'm going to do this with y'all. Let me zoom in just a little bit. Okay, so basically you have this word bank and I'm going to go through and label the plant cell with y'all, okay, just to make sure that you have these labeled correctly. All right, so first let's start with our nucleus. Okay, so it's going to be this blue structure. So we're going to do the whole outside. We're going to go back. Okay, so this is going to be my nucleus. Okay, so what is this little dark blue ball in the dead center? That's going to be our nucleolus. Okay, so then what is our free floating condensed DNA that is in the inside? That's going to be our chromatin. Okay, and now let's go ahead and do the easy structures first. Okay, what's my giant jelly bean on the middle of the cell called? That's my central vacuole. Okay, what about my two deli meat? So here's one deli meat and here's another deli meat. One of them's touching the nucleus and one is not. Which one touches the nucleus? The endoplasmic reticulum. So then we're going to have our rough and smooth ER. You can't really see it, so I'm going to go ahead and label this for you. But the red is going to be the rough ER and the purple is going to be the smooth ER. Okay, and then this arrow, this is a really bad picture, but it's pointing to a ribosome. Okay, and where ribosomes also found on the rough ER. Okay, so this picture is indicating right here, what is the inner dark green layer showing? This is our cell wall, so the cell wall of the plant cell that we're talking about. And now I don't really like filling in this answer box. I think that this is a cheap answer, but this is the wall of neighbor or the adjacent cell wall. Okay, so I'm not really concerned about labeling that one, but I did want you guys to know what that was talking about. Okay, so then next up, what is our like money for the plant? So this green structure is going to be our chloroplast. Okay, so then here's my powerhouse of the cell, so my mitochondria. Okay, so then what is the most inner membrane of the cell? So all cells have this, but not all cells have a cell wall. So what is this? This is going to be your cell membrane. And what's another word for cell membrane? Plasma membrane. Okay. So this little stick that is in here next to the other mitochondria and chloroplast, okay, this is going to be your centrioles. Okay, these are your microtubule filaments that are used during mitosis and meiosis. Okay, and then coming back to it, here's my other deli meat that's not connected to my nucleus. So this has to be my Golgi apparatus. Okay, so then what this arrow right here, sorry, is indicating the jelly-like fluid that fills the cell. What is that? That's the cytoplasm. Now, these two, I'm not going to quiz you on these two answer choices, but if you want to write them down, the plasmodesmata basically acts as holes in the cell wall that allows materials to transport in and out. And then this is a peroxisome. Peroxisome. Okay, and basically the peroxisome is going to act as like a lysosome, which is going to engulf waste and broken organelles or worn down cell particles. Okay, so there's your plant cell. Now I'm going to go on to the animal cell. Okay, so this is now page 7 of your interactive notebook. Okay, animal cell. So same thing, let's start with the big blue organelle in the middle. So this is going to be your nucleus. Okay, so then what is the dead center of the nucleus? Your nucleolus. Okay, and then what is your condensed DNA inside the nucleus called? Chromatin. Okay, so what are the noodle-like figures that help pull apart the microfilaments and microtubules? Okay, these are called centrioles. Okay, so then let's go ahead and do the easy organelles first. So what's my powerhouse of the cell? This is my mitochondria. Okay, so then I have my two deli meat, one touching the nucleus, one not touching. So let's do the one not touching first, is your Golgi apparatus. So then we know we're going to have the ER touching the nucleus. So one is rough, one is smooth. So what is the red one? If it has the ribosomes on it, that's going to be the rough ER. So that means the purple one is going to be the smooth ER. Okay, so then my really crappy arrow picture, this one is indicating a ribosome. Okay, so then this arrow next to the ribosome is indicating the jelly-like fluid that fills the cell. What is that called? The cytoplasm. Okay, so animal cells do not have a cell wall, but all cells have what? A plasma membrane. What's another word for plasma membrane? Cell membrane. Okay, so then centrioles help pull apart what microfilaments and microtubules. What are another word for those? Cytoskeleton. Okay, and then the last one that we have to answer, what is the animal version of a peroxisome? So what breaks down waste and other toxic materials or worn out cells is the lysosome. Okay, so there's your animal cell. So then the last thing that you have to do today is page 8, where you're comparing and contrasting animal cells and plant cells. Make sure that you list 3 similarities under similarities and 3 differences under each one. Okay, so I need 3 bullet points, 3 bullet points, 3 bullet points. Okay, and then the same for page 10, 3 bullet points, 3 and 3. Okay, great thing about being in the computer lab is if you still do not understand this, Google it. If you still don't understand it, message me, okay? So it is not terrible. Go ahead and knock out those interactive notebook pages. Don't forget that your test is tomorrow. Study the Quizlet. I'll send you an email with everything that's due tomorrow. But study for your test. Message me if you have any questions, and y'all are going to do great. Good luck. Miss y'all.